Welcome to this video mini lecture on the renal system, where we will explore the process of filtration, reabsorption, and secretion within the kidney, while gaining a better understanding of how blood is converted into urine. My name is Christopher Demas, and I'll be guiding you through these topics. Let me start by drawing the gross structure of the kidney. As you can see, there is a rich blood supply which allows the kidney to fulfill its purpose of filtering the blood. The pre-filtered blood enters from the renal artery, and the filtered blood exits from the renal vein. In addition, there are two structures of the kidney that can be visualized during gross dissection: the outer cortex and the inner medulla. Within each kidney, the functional unit that performs the filtration is called the nephron. Each kidney has roughly 1 million nephrons. Each of these nephrons span between the cortex and the medulla, as shown here. Since the nephron is a key structural and functional unit of the kidney, let's take a closer examination of its structure. The first part of the kidney where blood comes in contact with the renal tissue is called the renal corpuscle. This is the location of blood filtration and where the early filtrate is created. Filtration only occurs in this part of the kidney and nowhere else. The renal corpuscle consists of two parts: a cluster of capillaries, known as the glomerulus, and a capsule that surrounds the glomerulus that serves to collect the early filtrate, called the Bowman's capsule. And just to echo what I mentioned earlier, this is where filtration occurs. Let's go ahead and zoom in to view the renal corpuscle in more detail. Notice how the blood enters the glomerulus through the afferent arterioles, and the filtered blood exits through the efferent arterioles. On the outside of the glomerulus are the podocyte cells that contain small foot processes that cling to the glomerulus. The podocytes assist in filtration of the blood by generating thin filtration slits between the foot processes. Let's take a closer look at one of the capillaries within the glomerulus to understand how this is possible. Notice how the capillary has a number of layers that contribute to its selectivity in filtration. The early filtrate must be able to pass through the fenestrated epithelium, the basal membrane, and the spaces between the podocyte foot processes to be able to continue to the next part of the nephron. Something else to note that you should be able to identify is the key structures of the glomerular capillary bed in cross-section. Notice how the lumen is on the left, the fenestrated epithelium is the most interior layer, the complete basal membrane is in the middle, and the podocytes are on the exterior. Between podocyte foot processes are thin diaphragms, which further contribute to the selectivity of filtration. Let's now return to the larger view of the nephron. After passing through the renal corpuscle, the early filtrate passes into a series of twisted tubules called the proximal convoluted tubules. As you can see in the overlay of the nephron with the kidney, the proximal convoluted tubules can be found within the cortex of the kidney. In this area of the kidney, reabsorption and secretion takes place. The body reabsorbs key nutrients out of the urine that were removed from the blood, such as amino acids, glucose, and ions. In addition, waste products are removed from the blood through the process of secretion, such as urea. The proximal convoluted tubules are lined with simple cuboidal cells with microvilli. These microvilli are not found anywhere else in the nephron. On H&E slides, the proximal convoluted tubules may look fuzzy with a brush border epithelium, which is an absolute giveaway that you're looking at the proximal convoluted tubules. After this first pass of reabsorption and secretion in the proximal convoluted tubules, the refined early filtrate enters the loop of Hanley. There are various parts of the loop, including the proximal straight tubule, the thin descending limb, the thin ascending limb, and the thick ascending limb. The thick part of the loop contains simple cuboidal epithelium, and the thin parts of the loop contain simple squamous epithelium. This part of the nephron is only involved in reabsorption. Within the loop of Hanley, water and salt is reabsorbed. This means water and salt are selectively removed from the filtrate and returned back to the blood. The distal convoluted tubules are very similar to the proximal convoluted tubules in that they participate in reabsorption and secretion, and that they contain simple cuboidal epithelium. The distal convoluted tubules are, however, found more distal in the nephron, and they do not have microvilli, which makes their border look more sharp on H&E stain. Before moving on, I'd like to emphasize a specialized portion of the distal convoluted tubule known as the juxtaglomerular apparatus. This name, the juxtaglomerular, or JG apparatus, makes sense since it is found next to the glomerulus. Within the JG apparatus are two cell types, macula densa cells and JG cells. This structure is key in regulating blood pressure through the following pathway. The macula densa cells sense the low sodium levels or blood pressure and in response signal to JG cells to secrete renin. Renin is involved in the renin-angiotensin-aldosterone system, which acts to increase the systemic blood pressure. Do not worry about the details of this pathway though, as it will be covered in a future lecture. Next, the distal convoluted tubules merge with the last part of the nephron called the collecting tubules, and these collecting tubules converge to form collecting ducts. The urine can then be further concentrated in the ducts if necessary, and is regulated by antidiuretic hormone, also known as vasopressin. The presence of ADH promotes reabsorption of water, which serves to concentrate the urine. At this point, the urine passes through a few different structures prior to its elimination from the body. The collecting ducts eventually reach the center of the kidney, known as the renal pelvis, and then drain into the ureters. The ureters are muscular tubes lined with transitional epithelium. A picture of the ureters are shown here. Also, remember that there are two ureters, even though I'm only drawing one here. Next, the ureters conduct urine to the urinary bladder, which is the storage area for urine before it is ultimately eliminated. The bladder is also lined with transitional epithelium, which, if you remember from your epithelial lectures, can either be dome-shaped or flat depending on the amount of urine stored in the bladder. The last structure you should be familiar with is the urethra. As the urethra approaches the external orifice, the epithelium transitions from an uncommon stratified columnar to a non-keratinized stratified squamous type. Also remember that the urethra is much longer in males than females, since it must pass through the prostate and the corpus spongiosum of the penis. We covered a lot of information in this mini-lecture, so let's take a moment to review what we have learned. First, we looked at the kidney and learned about its macro growth structure, which includes the outer cortex and the inner medulla. We then discussed the functional unit of the kidney, the nephron, and how it fits within this macro kidney structure. We talked about the first step in forming urine, which is filtration within the renal corpuscle. We looked closely at the renal corpuscle at its components, such as the glomerulus and the outer Bowman's capsule. We then looked at the structure of the glomerular capillary with a fenestrated epithelium, a complete basal membrane, and a surrounding podocyte with many feed processes, which enables proper filtration. We then continued by following the early filtrate through the tubing of the nephron. First was the proximal convoluted tubules, which are responsible for reabsorbing nutrients and secreting waste products from the body. The proximal convoluted tubules are the only part of the nephron that has microvilli. Next was the loop of Hanley, and we discussed its function of reabsorbing water and salts. Third was the distal convoluted tubules. Their function is similar to the proximal convoluted tubules, however, they do not contain microvilli. The distal convoluted tubule also contains a specialized region called the juxtaglomerular apparatus that contains macula densa and JG cells that are responsible for monitoring and elevating blood pressure through the renin-angiotensin-aldosterone system when needed. Finally, we talked about the collecting tubules and ducts. These ducts are important for concentrating urine and are sensitive to antidiuretic hormone, which promotes reabsorption of water. The concentrated urine then passes through the ureters to the bladder, where it is stored until an individual urinates and it is eliminated through the urethra. I hope you found this video to be helpful, and feel free to review this topic as much as you need to. Thanks for watching.